Thank you, Ahmad, for a great overview. If we have time during discussion, perhaps we can talk about differences in design of master protocols between IO versus targeted therapies and any lessons that might have been learned. So the next speaker is Lisa Butterfield from the University of Pittsburgh, and she'll be speaking about incorporating biomarkers into clinical trials for PD-1, PD-L1 combinations.
Thank you. Well, greetings from Pittsburgh. Here are a few disclosures. So I wanted to bring to this talk the perspective of someone who's run an immunologic monitoring and cellular products laboratory for over the last decade, and who's dealt with a lot of institutional studies and multi-institutional studies. And so that's where some of these recommendations will come from. And the first, as Ahmad alluded to, is the need to have the right specimens saved under the right conditions. And even today, even with groups like Nordesis, who published, I mean, it was even a decade ago, of standard operating procedures to bank blood and serum to do immunologic assays, some of the groups that we've worked with, you can share SOPs, you can have conversations on the phone, and specimens will not be banked according to those SOPs. You will not get viable cells back, and you will not be able to answer questions. So this is pre-analytical variables still remain a problem, surprisingly enough. And I think, as we know now, guessing at a couple of immune assays at the front of your trial are probably not going to be sufficient to give you a big picture. So this was a great editorial piece from Nature Biotechnology that I think is still very important and relevant. And as was also alluded to earlier today, there are specimens banked in a lot of places. They're in a lot of freezers. I've been a part of a number of NIH grants that have tried to fund using the specimens that were banked to ask biomarker questions that are very challenging to fund. And then there's the issue of trying to do things in your own laboratory and share with your pharma colleagues who are trying to do their work in their setting to get the next biomarker tests approved. And also the assay, the new assay or the one assay you thought of, again, is not probably going to be sufficient. So I'll talk a little bit about the blood. I'm personally of the opinion that eventually we'll get back to the blood. When we know exactly what we're looking for, we'll be able to find it there. It's just as an average of everything going on immunologically in the patient, it can be hard to find it there at the outset. But you can look fresh, you can separate into plasma and serum. But what you get when you send out blood and you get that back to your laboratory, it can be hemolyzed, it could have sat around for a while, it could have been under the wrong conditions, in the wrong tube. And I'll tell you, for multi-institution studies, what we do is we send kits. And we pack our blood tubes in our kit and send those out so that there aren't just instructions, they're the actual tubes with the instructions that go to each of the clinic sites, so there are no issues. With tumor and with blood, serum and plasma, there are many things that we've been looking at for many years that are addressing the mechanisms of the studies that we're trying to do to answer the questions that we set out to ask. And there are a lot of very important questions, and some of these vary with the kind of trial you're doing. Cytokine trials, for someone like me who thinks a lot about cancer vaccines, did we activate our antigen-specific T cells the way we wanted to? In the setting that we're here for today for checkpoint blockade, where we're able to reverse immune suppression, where the target molecules on the target cells indeed activated. So the big picture is this a therapeutic improvement? And if not, why not? And so that's where I think we need to not just save the specimens to answer the question that we're asking when we design the trial, but we need to save the specimens that are going to answer the questions we think of while the trial's enrolling and the field has moved forward. And then when the trial is complete and the field has moved forward yet again, we're going to need more specimens very broadly banked to be able to answer all of those questions that we think of before, during, and after the trial design part. I'll remind you a little about CLIA. I think we know when we go and get our cholesterol test that no matter where we get it, we're going to get data and we're going to get the same number from all the labs because it's a CLIA test. It's very simple. And testing like this can be reasonably simple if it's one parameter, but it's much more complex when it's immunologic to get accuracy. What is an accurate measure of antigen-specific T cells? I'm still not sure what the gold standard is for that. Precision, reproducibility, to have reportable ranges, and to have the assay done in an environment that's also CLIA. Are your personnel competency tested for that assay? Is the equipment validated and monitored? Are the reagents tracked? So all of that can be a complex endeavor for getting the right biomarkers in your study. So here's the model that we settled on, and so this is how we've been supporting ECOG, Akron Cooperative Group trials. A patient is screened at any of the clinical sites. We prepare the kit for that trial. We put those tubes and vials and bottles and media and everything in a temperature-controlled kit in a box. We send the shipping box in an outer box. That goes to the clinic site. When the patient comes in, the blood is drawn, the tumor is sampled, everything's put, hopefully according to our instructions. Everything, we've created 2 temperature kits, so the serum can be at one temperature and the blood cells can be at another and the tumor is in another. All of these are shipped back to our central site within 24 hours. And of course, that's the concern. 24 hours have passed. So there's the lure to do things at your own site because it's fresh and you got it there that day. But then if it's more than one site, you've now introduced all of those differences between how your site and the next site does without actually having someone go and competency test each and every site to make sure that when they do it at that site, they'll be following the same instructions. So in our setting, everything is processed and banked within 24 hours of the blood draw, which means that no matter how many times we tell them not to draw blood on Friday, if the clinic is on Friday and that's when the PI sees the patients, the blood and tumors are drawn on Friday, which means we have to have people willing to come in on pagers on Saturday to process and bank those Friday samples. So we gather those data, we send those in for central analysis, and then in the fullness of time, the trial is complete. So can this work? This is functional. This is one of the most standardized assays out there. It's a single parameter, but it's interferon gamma production in an ELISPOT assay. So the top data set is from 2008 to 2009, 20 plates that we ran. The bottom is from 2002 to 2003. It was testing the same vaccine, and you can see the spontaneous interferon gamma production from the healthy donor control in each plate. It was 4.9 spots versus 5.4 spots per well, and the positive control 304 for the more recent and 284 for the other. I think those are pretty reproducible, which means in a standardized functional setting, you could actually compare those trials if no biostatistician in the room disagrees with me. And it did show that in this particular vaccine study that if you were in the group that did have a positive ELISPOT, you had a better median overall survival. But as you can tell, back then there were no tails, not appreciable tails on that curve. When we looked at non-functional assays in the same setting, we could see evidence for vaccination increased in certain markers on the T cells. MART1 T cells went up and they became somewhat more effector in phenotype. Flu cells as a control don't go up, which is good. We didn't vaccinate with flu, but those did not correlate with clinical response, so function over phenotype in these studies. I think it's also important to validate markers that other people have found in their studies. So ICOS ligand and ICOS upregulation on T cells found in other trials and other laboratories, something we could then look at in a multi-institutional study showing that on both CD8 and CD4 T cells, the patients who did better in this Ipilimumab versus Ipi plus GM-CSF study, the more ICOS on those T cells, the better. Of course, the tumor microenvironment is really the critical place where we're learning a lot of very important lessons, and I've been gratified to see so far in this meeting that a lot of us are highlighting all the same studies that we all find to be very intriguing and the leading studies and the leading biomarkers that we all need to be thinking about. Here for the immunoscore of Gallan and Friedman, this started as 3 markers in the tumor microenvironment in a cold tumor, colorectal cancer, and this went on to be validated, and here I'll put my SITC hat on, in a worldwide initiative to validate this biomarker in colorectal cancer, which was successful. But of course, while that validation study is going on in a 2 or 3 marker IHC, the whole field has moved on. And so this is now multispectral imaging, and you've seen several elegant examples of this. Here we're looking at melanoma tissue, looking at who's expressing the PD-1 and the PD-L1, T cells, myeloid cells, or tumor. Here we're looking at Tregs, immunoscore, and we're adding IDO to this to look at the architecture as well as the biomarkers. And so this is now the subject of another SITC initiative to bring together some of the leaders in this area to publish best practices and compare SOPs to, again, move this part of the field forward in terms of biomarker assessment. I think I'm actually the first person to show this slide today. So I think we're learning a lot about tumor mutations, and so I'll just echo some of the other studies that we've been talking about that are also biomarkers that have risen to the top in immunotherapy studies. And so that's mutation burden, a very important signal, not giving us as a standalone a cutoff in order to select patients yet, but very important data that we all need to be looking at and including in our studies. PD-1, PD-L1 expression, as others have highlighted, I'll just echo that this stains differently and works differently between different tumors in different clinical settings, but it's also absolutely critical. We can't do a study without looking at this. Here, the biomarker that crosses the tumor type barriers, the mismatch repair deficiency. This shows all the different tumor types and their frequency of being mismatch repair deficient, showing that this is a true biomarker that can be incorporated across different tumor types and is also critically important to be measuring in order to validate this and identify the CLIA assay that we can use for this. And we've also looked at tumor microenvironment gene expression assays. So this is also, I think, something that keeps bringing up the same themes that we're all, again, talking about. Interferon gamma, cytotoxicity. Here's the figure that you have already seen today, looking at the chemokines and the CD8 and all these other markers that, again, all of these themes that are cross-cutting all of these trials that give us a core set of biomarkers that I think we need to include in all of our studies. So this also is giving us directions to go when we don't see the right kind of tumor. So when we don't see the inflamed PD-L1 high, heavily mutated tumor, what are we going to do about it? I'll tell you, make a dendritic cell-based vaccine, but where there's really good data is looking at T-VEC injection. So here's showing that T-VEC injection is turning the cold tumor hot and creating PD-L1 protein expression, interferon gamma gene expression profiles, and something that we can build on then in combination studies. So I'll finish up by highlighting, again, putting on my SITC hat. The Biomarkers Task Force met some years ago, just as the field was beginning to take off, to make some recommendations for incorporating biomarker studies in trial design. And there, I think our point was to highlight that we should control the variables that we can control, and that's to make sure the specimens are processed properly, that the assays are conducted under SOPs, and then to make sure we're saving specimens to understand all the variation between patients that we can't control. And that's the tumor mutations, the patient genetics, and to make sure that we had specimens for all of those things. Since then, the field has really moved forward, and just so everyone knows about these resources, we have a series of white papers looking at the hurdles and ways to move forward. The standardization and validation task force had such a big job in front of them. They had 2 white papers to talk about analytical validation of biomarkers and then clinical validation of biomarkers, because, again, in the world we live in today in immunotherapy, we're not looking for BRAF mutant or not. We're looking for biological cellular mechanisms that are much more complex to standardize and validate. There are also the wonderful new technologies that we've been hearing about. And the new SITC task forces are around data sharing, we'll be talking about later in this meeting, immunoscore image sharing, and again, multispectral imaging. So the other things we need to think about in trial design and incorporating biomarkers, it used to be that all immunologic biomarkers were exploratory, and now they're both integral and integrated, thinking about making decisions about where to put patients for treatment based on the assay outcomes. And remember, of course, integral biomarkers require that CLIA-certified laboratory in that highly regulated environment. And the cost, one of my internal PIT investigators said that they wanted to have a cytokine, a serum cytokine, as a entry measure. And so could we make that CLIA, please? Because it was simple. It was just an ELISA. And we gave them the bill for what it would take, for just our cost to run all of those controls and same assay over days. And it didn't happen because they couldn't fund that with a grant. So that did not get a CLIA certified and an enrollment criteria for that study. So I guess to summarize, my recommendations are to, of course, focus on tumors for now, but keep the blood, keep validating biomarkers as we've heard about today that have given us these great signals, but also include those high throughput hypothesis generating assays and specimens, because that's going to answer all the questions that you have at the end of the trial, because the field will have changed so much. And I think sometimes we're all holding on to different parts of the elephant. We're looking at multiple tumor antigen-specific T-cell responses, which I think is a measure of determinant spreading, which is a measure of the mechanism of in vivo cross-presentation, which requires those cross-presenting dendritic cells. To catch that in action is probably a tertiary lymphoid structure that is actually doing the cross-presenting at that time, and that is aided by a high mutation load in an interferon gamma signature containing tumor. So I think all of these things are singing the same biological song about the tumor that's going to respond to these therapies. So I will stop there, and thank you very much.